Good morning, friends. So, in last 2-3 lectures, I am telling you about the different modified Mendelian inheritance. And today, in this lecture, I will be discussing you about the modified Mendelian inheritance. But here onward, there will be two or more locus involvement. And in this series, today I will be starting with gene interaction. Gene interaction. So, what is gene interaction? So, let us start that what is gene interaction. So, in gene interaction, there the genes which are present on different locus, they will be interacting with each other and they will contribute for the determination of single phenotypic characteristics. So, for controlling one characteristic, genes or alleles present at different locus, they will be interacting with each other. So, we know from Mendel's law of inheritance that genes assort independently, but it is not always true. At that time, when I was discussing you about the law of independent assortment, at that time also I had told you that this law is not universal. So, what will happen? They will not act independently in their phenotypic expression, and instead of that, the effect of gene at one locus will depend on the action of genes or alleles present at another locus. So, this will be known as gene interaction. So, it was discovered by William Batson and Reginald Punnett. So, Batson and Punnett discovered it when they were working with the inheritance pattern in comb pattern in poultry. So, inheritance pattern of comb pattern in poultry. So, we will see how this comb pattern in poultry is being controlled. So, the products of gene at different loci combined and they will produce new phenotype which will not be predictable from the single locus effect alone. So, as in the case of single monohybrid cross, we were able to predict the F2 genotypic or phenotypic ratio, but here since two alleles at different locus are involved, and we were thinking that okay, the comb pattern poultry is being controlled by may be controlled by one gene. So, if we thought that it is controlled by one gene, we will not be able to predict the actual comb pattern inheritance. So, for that, the work done by Batson and Punnett, they come with the finding that this is controlled by two different genes present at different locus. So, common example of gene interaction is the comb shape in chicken. So, what is comb pattern? What is comb actually? So, you might have studied this comb structure of comb in your LPM classes, but just a definition: the comb is fleshy structure found on the head of the chicken. So, there may be different types of comb pattern. You definitely might have studied in LPM classes that there are single comb, pea comb, walnut comb, rose comb, etc. So, it is considered that genes present at two loci, that is R loci and P loci, and different alleles, different forms of two different forms of the two genes interact to determine the four types of combs that I had told you just now. Now, what are those four types? The four types, the first one is walnut comb. So, what is walnut comb? So, for understanding walnut comb, first we will see what is single comb. Here on the fourth number you can see the single comb. So single comb is produced when a recessive allele is present at both the loci. So now it is clear that it is controlled by two loci, alleles present at two loci. So when all the four alleles present at two different loci are in recessive form, there will be single comb pattern. Similarly, the rose comb, when there is one dominant allele present at one locus, that is R locus, and at other locus, that is P locus, both the alleles are in recessive condition. In that condition, the comb will be rose comb. Rose comb. So here you can see that one is capital R, another it may be capital R or small r, but at different loci, second loci, both the alleles will be recessive one, that is small p, small p. Similarly, in case of P comb, the alleles present at P locus, they will be in either dominant homozygous or heterozygous, but alleles at R locus, they will be in the recessive homozygous state. Then the comb pattern will be P comb. So you can see the morphology, but I am interested in discussing about the genetics of the comb pattern. For morphology, you can see any of the poultry book or your LPM notes. Then what will happen if there will be dominant alleles at both the loci, that is at R loci and at P loci? So in that case, the expression will be codominant. That means a new phenotype will appear, and that phenotype is the walnut comb. Walnut comb. So for being walnut comb, at least one alleles at both the loci should be dominant one. So this is about the genetics of comb pattern in poultry. So why this happen? If you see here the inheritance pattern, so first is the rose comb. Rose comb, you can see the alleles at both the alleles at R locus are dominant and both the alleles at P locus are recessive. Similarly, the P comb, when they cross together, the appearance was walnut comb, that means codominant. But when this codominant, that is walnut comb, were mated with the walnut comb, in F2, the ratio was not changed. That is, the ratio was 9:3:3:1. But the combination, as I discussed earlier, the combination was depending on the dominant or recessive status of alleles present at these two loci. So the important difference we have here, that we have four distinct category of a single trait. So in case of there was four distinct category in Mendel dihybrid cross also, distinct phenotypic category, but there we were studying about two traits. But here there are four distinct category for single traits only. So this is the basic difference in the gene interaction and simple dihybrid cross, simple Mendelian dihybrid cross. So it is considered that why these four distinct classes for single trait. So it is considered that it may be because of the interaction between the products at the biochemical level or cellular level. That means these two locus interact together in how they will interact. They will interact through the product of the biological or cellular level. So this is about the comb pattern inheritance in the poultry. This is maybe the pattern inheritance pattern, different inheritance pattern is due to the gene interaction, and this gene interaction is different from the epistasis that we will be discussing it in the next class, and the simple Mendelian dihybrid cross, and what is the difference that we have discussed in this class that you should take note of. So thank you for listening. Thank you. Thanks.